Problem. Okay, I'm joining a class now, so we'll talk later.
So are you? So are you? Then you the news. No, small fun about time coming.
Hello, good evening, everybody.
Good evening.
How did you spend your weekends?
It was great, Grandpa.
Good. All right. So we are here to continue. And of course, we start week 2, but still on module 1. So we say week 2, module 1 continued. We hope to finish with module 1 by tomorrow. And then start with the second module, still on andrology, and that will be andrology 2. And hopefully that will be on Thursday, because we do 3 days in a week, Mondays, Tuesdays, and Thursdays. So if by the grace of God we are able to complete the module 1 tomorrow, then we will begin the module 2 by Thursday, God willing. Now, before we start with this week's session, if there's anything that was discussed last week, which you may want to seek better and further clarification, please do so. I've said over and over again that this course, you should be enabled. You should have that kind of capacity to understand exactly what you're doing, not just rushing through and reading through. And there are a lot of practicalities, not practicalities as in terms of laboratory practicals, but you should be aligned, whether you are male or female, you should be able to align with some of the things that we are discussing with our day-to-day practice. All right, so let's try to be interactive by asking questions, by bringing on board what you know already, and for which you may want to seek more clarification. Of course, scientific clarification on some of the things that you know on our day-to-day lessons. If there's nothing to discuss, then we move on to the male accessory organs. Of course, male accessory sex organs, and accessory means a helping. It means that the main work will be done by somebody and something else is also supporting it to make it complete. All right. And so, of course, you know that now the major work will have been done by the testes. The male reproductive system is basically for procreation. Procreation is producing a section such as the spermatozoa, which will have to be transported onto the female reproductive tract for it to engage in fertilization and for that matter, reproduction. Okay. So, and that work will be done by the testes. Again, you also need hormones, the androgens, and this one, testosterone. And you've all come to know that testosterone is also produced in the testicles, of course, by the lady cells. All right. So if these are the major roles of the male reproductive system, and they have all been completed by the testes, then it means that any other organ associated with the male reproductive system is just providing some kind of supporting stuff. All right, that's why you consider them as accessory, because the major work has been done by the testes. All right, so now let us look at the male accessory organs. What constitute accessory organs? So this is a collection of glands and ducts that contribute to the formation of semen and facilitate sperm transport. As you move along, you know by now that sperm alone doesn't constitute semen. All right, just as we have the blood. The red cells alone doesn't constitute blood, even though we see blood as something which is red, and therefore it's basically red cells, but of course, no. All right, so the other things that make up blood. So if you transpose that into the semen, yes, you talk about the sperm. The sperm alone doesn't constitute semen. So as you move along, you see the commonalities of blood and semen. All right. For example, you know that once you have blood and you sperm, you separate it into the cellular portion and the fluid portion. The fluid portion becomes the plasma or serum, as the case may be. And then the solid portion becomes your cells, blood cells. Same can be said of semen, to the extent that semen can be centrifuged, just as we do. And then once you centrifuge, you get a fluid portion and then a cellular portion. The cellular portion now constitutes the sperm cells, and then the fluid portion becomes seminal plasma. All right. So there's that kind of semblance. All right. So, and you deal with that as you move along. So I'm saying that the major aspects of the male reproductive system is the work is actually done by the testes with respect to the production of the sperms and also the production of production and secretion of the androgens.
I don't know.
Is the testosterone. Now, all other things that add up to it become accessory. I think that point is well. And then, if you look at the way, please, can you mute, please? If you look at the male reproductive system again, and the way we have tackled it, from the scrotum, from the penis, from the angle of the testes, we are moving up. All right. And so after testes, we've done the epididymis, we've done the vas deferens. And so now we come to other things. The other things that make up or constitute what is generally classified as accessory are the seminal vesicles, the ejaculatory duct, the prostate gland, and the bulbourethra, which is also called the Carpus glans. All right, so we're going to tackle them one by one. And once we finish, you also see the role each of them play. So that once you understand this aspect, like I said, if you go to seminology, you see that you understand the aspect of semen formation, everything from the basis, and you appreciate the other things that come along with it. So let's have an overview of the male reproductive system as we tackle with the accessory glands. Right, so this is the overview. So we said that the accessory organs, we're going to talk about the seminal vesicles. And this is, if you look at the arrow, this is the seminal vesicles. We've done with the testes, the epididymis, the vas deferens. Right. So the vas deferens, as we move along, you see more clearly that the terminal, when you say terminal, that's the end. So probably it is inappropriate to say terminal end, because the terminal is the end. So at the terminal stages, the terminal portion of the vas deferens, you see that it is enlarged. And I've told you that enlargement, that enlarged portion is called the ampulla. So you can call it the ampulla of the vas deferens. So, and these are seminal vesicles. All right. So we are going to look at the seminal vesicles, and because it is a gland, every gland must have a duct. Glands produce or secrete fluids. And as they secrete the fluid, there must be an outlet. That outlet from a gland is the duct. So every gland will have a duct. All right. So as we have this as the gland, then the end here becomes the duct of the gland. All right. So this is also the vas deferens. And the end here is enlarged to form the ampulla. So the ampulla will now join up with the duct of the seminal vesicles to form another structure called the ejaculatory duct. This ejaculatory duct will now open or cut or traverse the prostate, and this is the prostate. So into 8, every gland will have an opening. So this gland, the duct here and the duct here are now also becoming another duct, which is the ejaculatory duct. They will also cut into the prostate and open into it. Remember, it is the same region where you also going to have the bladder also opening into it. So it plays that critical role. The prostate plays that critical role. And then from there, you are going to have the bulbourethra, because this is urethra, and this under gland. So these are and they all make contributions to the formation of the semen. So we have the sperms, which are being produced here and being transported via the vas deferens and coming here. So from here, you are going to get some additions, additives, fluids that are mixing up with the sperms to now create the semen proper. Once that happens, then it can now come out in the form of seminal fluid. All right, so these are the things that we're going to talk about. seminal vesicles, the ejaculatory duct, the prostate, bulbourethral glands. Once we finish with that, it means that we are finished with the entirety of the male reproductive system. So this is another picture of the male reproductive system from another perspective. Of course, you have the penis, the testes, the epididymis, the vas deferens. Look at it here. It's very, very important. And as we move along, you see that this vas deferens becomes a conduit. I mentioned conduit. All right, so this conduit can be severed or cut. All right, once it is cut, then it means that sperms cannot be transported to the outside, and that is vasectomy. So we will deal with that later on, but I just want to know that sperms can be produced, everything down here is being transported here, and then if you want to stop it from coming out entirely, then you must do something about this duct. Okay, so this duct can be severed. When I say severe, it can be cut or can be tied. And once it's tied, now it becomes an obstruction. So sperms can no longer come out. Remember, as we move along, like I've said, this is another perspective of the male reproductive system. Now, as this duct comes here, you see that this is the end of this duct, and it's enlarged, and that's the ampulla. All right. So the ampulla, this is also the seminal vesicles, they join, you get the ejaculatory duct, and so forth and so on. Now, I've told you that if there for some reason a person accumulates sperms for a very long time, the sperms are being produced. As you move along, you see that sperms are produced every day. In fact, every minute, every second, sperms are being produced. And within the epididymis, they are stored. And we learned that the maturation starts within the tail, within the head, move through to the body. By the time it gets to the tail end here, it will have completed the maturation, and then they will store. But if you store them for a long time, see that occasionally you get some pushing themselves up. Once you push themselves up here, they come to the ampulla, the sperms, even though without sexual activity, because they have been too much produced. Then the sperms will be, you can get some sperms here. All right, getting also, and know that this ampulla also joins with the ejaculatory duct. And once it moves into the ejaculatory duct is also a union of the ampulla, the ampulla of the vas deferens, and the duct. So of course, once sperms are now coming from here and moving to this, and this is also like a storage organ. So you see that some of the sperms will move into the ejaculatory duct, and some will push up. Of course, some will push out. Like I've said, if for a long time you have not ejaculated, you see that sometimes you are urinating and then there are sperms inside. All right. Some of the sperms will come out. Some will also move backwards and then get into the seminal vesicles. You know, the seminal vesicles is becoming like also another storage organ. So you get sperms being stored here. That doesn't mean that the vesicles also produce sperms. They don't produce sperms. But because there have been massive accumulation here, some have been able to move up here and then be locked here. So as we move along, I just want to use the opportunity to also enlighten yourself so that once it gets to the real stuff, it becomes easier to understand. For example, if there's vasectomy, okay, if there's vasectomy, somebody undergoes vasectomy, and let's say after some time, after a week or so, he goes to have sex. And then the woman gets pregnant. And then the woman says, no, I'm not responsible. I've done vasectomy. And once you do vasectomy, it's like you have become sterile. That is the way any man, if you don't want to give birth to children or something any longer, the best way is to get vasectomy done. All right. So if you do vasectomy, and like I've said, once you get there, you throw more light on it. You don't say that you have done vasectomy and therefore the following week or so you are okay, you go and have sex, and then the woman gets pregnant and you say it is not, you are not responsible. You are, because like I've told you before, at any particular time, there is some spillage of sperms that comes here and being stored here. Okay, once they are stored here, you have severe dispersion. Sperms are no longer coming. What about the sperms that have been locked here? So it means that if you have sex post vasectomy, you are likely to also, because semen is a mixture of the sperms plus other fluids. All the accessory organs are functioning once you have vasectomy. All other things are working. So you also have sex, you can get ejaculate, and you can feel everything that a man must feel. So the seminal fluid which is coming out is not devoid of sperms. So what it means is that there should be a certain time span, or after a certain number of ejaculates, before this place will be devoid of sperms. All right. And you deal all this because as a scientist, sometimes you can be filled with some of these challenges. Somebody will come to you, can come and explain something to you. Master, this is what I've gone through. This woman is saying that I'm pregnant. You should be able to have that scientific information to be able to give the person the right information. All right. Good. So this is still an overview of the male reproductive system. So you're going to now take them one by one. The seminal vesicles. So it's also called vesiculae seminalis, or seminal glands, or vesicular glands. So I'm bringing out all this so that sometimes
this one for courage. Once you finish it and then you use all free sleep.
Miriam, will you put your mic off?
Sorry.
Okay. So we have various names for certain structures or organs. And once you are going through a comprehensive study of the of male reproductive system, you should be familiar with all these things so that don't say, oh, there's difference between vesicular seminalis and then let's say seminal glands. Okay, seminal glands because it's a gland that produces or secretes hormones. All right. So there are paired glands found in the pelvis. This is basic information, and they are specifically located near the base of the bladder, above the prostate gland, and in front of the rectum. Okay, so this is giving you an idea about where it is located. All right, the duct of the seminal vesicles. Remember, I've said seminal vesicles is also a gland, and every gland produces fluids, and that fluid must come out, and the tract or the pathway for the excretion of its fluid is a duct, whichever, whether it is prostate or whatever, for as long as it is a gland, it must have a duct. All right, so the duct or the seminal vesicles combined with the ampulla or the vas deferens. I've said this already. And the vas deferens, the ampulla or the vas deferens, we're talking about the enlarged terminal portion of the vas deferens to form the ejaculatory duct. I think this is also very important, which then empties into the prostatic urethra. All right. And again, this is the seminal vesicles. So the description that we've said is, first and foremost, is within the pelvic area. All right, this is your pelvis, the pelvic area. So it is one of the organs that lies within the pelvis. All right, and also behind the bladder, you notice the bladder, behind the bladder, and also above this is the prostate, so above the prostate, just that description. Okay, so it's located near the base of the bladder, above the prostate gland, and in front of the rectum. Okay, so that is just to give you better and further information about the location of the seminal vesicles. There's also a broader picture about the seminal vesicles. All right. And it is important to note here that this is the seminal vesicles, vas deferens coming all the way from the epididymis. All right. And as it ends in an enlarged portion. And what is this portion? You can tell me. This portion or the vas deferens is what?
The ampulla.
The ampulla. All right. So the ampulla will now join with the duct of the seminal vesicles. Okay. And then they enter into the prostate. Okay. So it is something that you can't miss. The same thing that is also being amplified here. All right. Good. So the gland has a lobulated structure that is in lobes with inner mucosal lining of simple columnar or pseudostratified columnar epithelium with goblet cells. Wherever you have goblet cells, what comes to mind?
Secretion.
Secretions. All right. So this is a secretory organ. All right. That's all. Good. So look here, the columnar cells of the seminal vesicles are highly influenced by androgens, which are testosterone. All right, so before they can secrete, they need the support. Okay, they need to be stimulated by testosterone. So you see that testosterone is very, very important within the male reproductive function. Okay, so under the actions of the testosterone, the seminal vesicles produce and secrete. They produce and will secrete yellowish, viscous fluid called the seminal vesicle secretions into the forming semen. What is happening here is that we have the sperms which have been produced elsewhere and being carried along. Then you have the seminal vesicles, which is also producing a yellowish, viscous fluid. All right. So they will also mix with the sperms. So this will tell you that as we move along, you will see that the seminal vesicle secretions, when it comes to the volume per se, it constitutes about 60 to 70% of the semen volume. So if it's yellowish, all right, if other portions which are of different colors, let's say colorless, mixed up with something which is yellowish, what will happen is that at the end of the day, the semen will never be colorless. It can't be colorless because this secretion from the seminal vesicles, the volume is about 60 to 70%. So the color, which is yellowish, can only be diluted a little bit so that you will never get semen which is colorless. Okay, so that is also the idea here as we move along. So seminal vesicles may store sperms. All right. We see that may store sperms, not that they will have to store sperms. As I've said earlier, it happens especially where there has been a prolonged abstinence. Where there has been a prolonged abstinence, the tendency is that sperms will move up and some of them will be stored in the distance. So it's not like a situation that you say that the sperms will automatically be stored in the seminal vesicles, which may be the reason for continued sperm in the ejaculates of men after vasectomy. I think I've explained this already, that because sperms may be stored within the seminal vesicles, the chances are that even after vasectomy, the person, anytime that individual ejaculates, there's a tendency for sperms to be part of the ejaculate, and therefore the likelihood for fertilization as such an individual. So this is still the same picture you saw earlier. And then the muscular tissue inside the seminal vesicles pushes fluid out of the glands and into the forming semen. The semen is now being formed, okay? And this seminal vesicles also plays a role by producing fluid that will mix up with the sperms for the formation of the semen. All right. So we talked about the seminal vesicle producing fluid. So let's see what is in the fluid. The secretions of the seminal gland, I've said that seminal gland is the same thing as seminal vesicles, contribute significantly to the overall volume of the semen. We've said this already. The fluid ultimately constitutes the major fluid portion or the major fluid volume of the ejaculate. When talking about ejaculate, we are talking about the whole volume of fluid or semen which will come out during sexual intercourse. I mean, and then during ejaculation, that volume that comes out is called ejaculate. Now, if you take that as a whole, then about 60 to 70% portion will have been coming from the seminal vesicles. All right. It is important to have all this basic information because if that's the case, then if somebody produces semen and the volume has a low volume, you realize that where do you have to point your finger at? Yes, somebody produces semen and what is coming out is of a low volume ejaculate. Where will you first and foremost suspect to have a problem?
I think it will be a problem with testosterone production.
The
testosterone production, since it is also.
No, I'm saying that you have produced semen. All right. You have produced semen. The volume is low. Where do you think will be your first suspicion?
The seminal
vesicles.
Yes.
Because the entirety of the volume, you are supposed to produce between 60 to 70%. So if you default in your contribution, then the entire volume will suffer. All right. So that is why before you now think about what could have also be happening to the seminal vesicles and blah, blah, blah. All right. So all this little information will make you appreciate your semen analysis better with science behind your knowledge. Now, we'll also talk, like I've said earlier, we are going to talk in details about all these things. Now, however, the secretions from the seminal vesicles are mainly included in the late ejaculate fractions during ejaculation. What is happening here is that during ejaculation, the other day I mentioned that during ejaculation, the fluid comes out in spurts, S-P-U-R-T, that like in fractions, it will come portion after portion. So the first portion, you should know where it is coming from. The second portion, you should know where it is coming from. Then later portion, you should know where it is coming from. So if you have semen or you are ejaculating, now the first portion of the ejaculate doesn't have much of the seminal vesicle secretions because they form the major part of the latter fractions. I hope that point is also clear. You should understand all this so that as we move into the seminology proper, it becomes easy to understand the whole concept. We are saying that the seminal vesicle secretions form the major fluid volume. All right. However, because during ejaculation, the fluid comes out in fractions. We are saying that the first fraction doesn't contain much of the seminal portion. The seminal fluid portion is not part of the first fluid that will come out. As we do the other aspect of the seminology, you see, okay, then which fraction is the first, which fraction is the second, which fraction is the third, and so forth and so on. We will do that in detail. But I'm saying here that since you're on seminal vesicle secretion, I'm just saying that during the ejaculation, the portion that comes out last contains much more of the seminal vesicle secretion than the first and second portion that came along. All right. Seminal vesicle secretions, or the fluid, the secretions in fluid, are rich in fructose, prostaglandins, and proteins, including seminogens. All right, seminogens. So seminogens are a certain kind of protein. So we're also going to deal with all these things one by one so that you appreciate them. The seminal vesicle secretions contain a lot of fructose, prostaglandins, and like we said about the epididymis. If you know some of the constituents of the fluid that they produce, then those constituents, some of them can be used as biomarkers. Is that not true?
Yes. Yes, please.
So you are doing the semen analysis. At the end of the day, you will be able to determine which aspect or which portion or which structure or which organ of the reproductive system has a problem, so that you fault everybody. Like it said the other day about the urine. If you have RBCs in urine, you should be able to have an idea about where the rhesus are actually coming from. And once you know that these rhesus are likely to come from this portion of the urinary tract, then you know how to also make specific diagnosis. All right. So similarly, if the semen has a problem, you should be able to, you don't have the facility to do this test ABC. But if it were in elsewhere, we can do this test, we can do that test. But if you don't know it, then even if the doctor makes a request, you say, what test is this? All right. But once you have that knowledge, you can even recommend to the doctor, can we do this test? We don't even do it here. But I know, and I've been made aware that a certain facility this way does this test. Can you go and do this test to also support your diagnosis? Then you are making positive contribution to the management of the patient. All right. So coming back, we see that seminal vesicles, they are rich in fructose, they are rich in prostaglandins, they are rich in proteins, including the seminogens. All right. So what of the fructose? If they have fructose, what is it? The fructose serves as the one talk about the carbohydrates. All right. So the fructose serves as the energy source for sperm cells. So the sperms need to survive. They must have an energy source. And the energy source is derived from fructose. And these fructose are produced by the seminal vesicles. Okay, so if you have seminal vesicles have a problem, then sperms will come out and they are dead because they don't have energy to survive. All right, so these are some of the kind of things that you should know so that it helps you to understand your semen concept far better. All right. So again, you see the seminal fructose. When I say seminal fructose, you're talking about the level of fructose in the semen. The main, and I've said that this is the main sugar related to metabolism and sperm motility. And known as sperms will move. Once they move, they need a lot of energy. All right. And their energy source is the fructose. All right. So the seminal fructose now becomes an important biomarker for the performance of the seminal vesicles. I hope that point is okay. So that determination of seminal fructose may provide an indirect evidence of seminal vesicle dysfunction. So that what it means is that if you have the facility to determine the level of fructose in a semen, it can tell you whether the seminal vesicle is functioning properly or not. All right, so that is the concept. All right. Again, the seminal vesicle secretion is also an alkaline in nature. All right, so the seminal vesicle fluid is alkaline in nature. Of course, they have high pH, which helps to neutralize the acidic environment of the male urethra and the female reproductive tract. You see, we've said that the seminal vesicles portion or the fluid portion or its contribution to the semen is as much as 60 to 70%. And remember, I said that because of that, the color is also yellowish, and therefore that explains the grayish coloration of semen. All right. Now again, it has the high pH. If it has a high pH and its proportion is 70%, 60 to 70%, that's a high pH. Even if there's another fluid coming from elsewhere, which is mixing up with the semen, because the semen proportion, the seminar vesicle proportion is higher, and its pH is higher, it will automatically give you an overall alkaline composition. Are you following the discussion, please?
Yes, please.
Yes. So, and why is it important in this way? It is important because, we said that the penis, and for that matter, the urethra is urogenital. It's playing a role in the urinary system. It's also playing a role in the genital, that's sexual aspect. Remember that urine is naturally acidic. Urine is natural as if a urine is becoming alkaline, it means that there is an infection. See, and this is automatic. The urine, you have a lot of organisms that may be splitting urea. Urine is made up of urea, a high composition of urea. And then if you have a lot of organisms splitting the urea, what do you think will happen? So you are going to get ammonia as the end result. Ammonia is alkaline. Okay, and so you are going to get a lot of, the urine becomes alkaline. All right. So what we are saying is that the urine is frequently passing through the urethra. So what do you think will be the pH status of a normal male urethra? Who can tell me? What will be the acid-base balance, or let me put it that way, of the male urethra?
It will be alkaline in nature.
No, it will not. It will not. You know why? Because the urethra, more urine passes through the urethra more than anything else. How often will you ejaculate? But the day you may pass about 3 or 4 portions of urine, and the urine that you are producing is naturally acidic. And I'm saying that if your urine is alkaline, straight away tells you that there's an infection. All right, straight away tells you that there's an infection. Otherwise, a normal urine should be acidic. And if the urine is normally passing through the urethra, then it means that the pH of the normal urethra will be acidic. You see, the normal pH of a male urethra. Hello. Alex and Joseph, will you please put your Alex, don't please put your okay. Coming back, I'm saying that the seminal vesicles secretions is alkaline in nature. Okay. And because it contributes about 60 to 70% of the entire semen volume, and that portion is alkaline, it makes the overall pH of semen to be alkaline. That's also, you should note that. Okay, so that even if a proportion from the prostate or seminal vesicles from other aspects are acidic, because the seminal vesicles contribution is higher and it is alkaline, it will make the total overall acid-base balance to be alkaline. That's why a normal, for example, when we get to the semen, you see that a normal pH for semen is actually between 7.2 to 8. Once it is more than 8, there's a problem. Once it is also less than 7.2, there's a problem. Even if it is 7, that is neutral. There's a problem because it should not be neutral. It should be alkaline. And once the pH is, it means that the proportion of the seminal contribution is becoming weaker. Okay, so those are some of the aspects that you should know as scientists. All right. And I'm saying that why is this important? It is important because sperms, as they are, sperms cannot survive an acid environment. Sperms will die within a matter of minutes in an acid environment. All right. So if they are going to pass through the urethra, and the nature of the urethra, like I said earlier, the nature of the urethra, the tract is acidic. Why is it acidic? Acidic because urine is also by production, urine is automatically acidic. So if urine is passing through the urethra all the time, then automatically it will leave traces of the urine along the tract of the urethra, and therefore it will make the urethra acidic. Meanwhile, semen, which is coming, the sperms in them will be killed by acid. So what must happen is that the contribution of the seminal fluid should be strong enough to make the entire seminal fluid alkaline, so that at the end of the day, they will go to neutralize the acidic tract of the urethra. I don't know, please get this point very, very well. All right. Again, the vagina is also automatically acidic. Who knows the pH of the vagina? Who can tell me the pH of the vagina?
5.0.
No.
Around 3.4
Yes,
to 4.
around 3.4 to about 4. If a pH of 4.5 in the vagina is said to be alkaline. The pH of the vagina is so strongly acidic. That's why you can only get candida there. You know, candida is candida is also like a fungi. And you know that fungi, if you are going to, if you look at the media for in the microbiology, if you look at the pH of media, most of them are around 7.2, 7.2, 7.2. But if you talk about SDA, sabrodestrost agar, that is around 6. That's acidic. And just because sabrod is being used to culture fungus, and therefore candida will grow very well on sabrod than anything. All right. So they need an acidic environment. That's why candida, for example, can feel comfortably within the vagina and survive there. Otherwise, all pathogenic bacteria cannot survive in the vagina because they cannot stand the pH of the vagina, and you have the lactobacilli producing a lot of lactic acid to maintain the acidic environment of the vagina. You understand? So, but this is the place that this thing is going. The sperms are going. The sperms are going into the vagina. The vagina is highly acidic, and sperms will be killed by acids. So what must happen is that the sperms must find themselves in a fluid which is highly alkaline to be able to neutralize not just the tract of the urethra, but also because at the end of the day, we are interested in reproduction. Is that not it? We are interested in the sperm going to fertilize an egg and then listen. So if you don't protect the sperms, what will happen is that they will all be dead within the vagina, because the vagina is naturally acidic, and the sperm too cannot survive an acidic environment. So you need an alkaline semen to be able to neutralize not just the acid of the urethra, male urethra, but also the vaginal environment. So if the seminal vesicles fails, I'm afraid you are heading towards infertility, because you may be producing sperms all right, but first the sperms must be able to live, okay, by having energy source, which is the fructose. And it is the seminal vesicles that are producing that. The sperms they feel comfortable in an alkaline environment. And it is the seminal vesicles which produces that environment. So you see that if you have your seminal vesicle dysfunctioning, you are likely moving towards infertility of a sort. So that is the critical role the seminal vesicle secretions are playing. So here say that this alkalinity is essential for sperm survival, as sperm cells are sensitive to acidic environments. And we told you that the acidic environments, first and foremost, even the male urethra itself is acidic, because the male urethra, like I've said this over and over again, I want you to note that the male urethra is technically acidic because of the urine passage all the time. Normal urine is also automatically acidic, and therefore it makes the urethra acidic, period. The vagina is also very, very highly acidic. Okay, remember your bro. I am the acid near. So you see an acid environment at 3.4, it's not easy. So God has made it easy for us to be able to pass through and then survive. The sperms will not survive. Anyway, that's just by the way. So you need that alkaline environment for the protection of the sperms, for their motility, for the everything. So that is the key role of the alkalinity of the seminal vesicles. Good. Then there's also prostaglandins. The prostaglandins has a very unique history. Previously, it was thought that, previously, it was well noted that semen has prostaglandins. That one, there was no problem about it. But it was thought to be coming from the prostate glands. Hence the name prostaglandins. That is the prostate gland that is producing this kind of entities. So that's why it was previously called prostaglandins. Even though the name is still there, but it tells you that it was abundantly clear that semen contains prostaglandins. And this prostaglandins was thought to be coming from the prostate glands. Hence the name prostaglandins. However, it is now abundantly clear that the prostaglandins in semen are actually coming from the seminal vesicles. I hope that point is clear. Right. So the fact that prostate gland was thought to be the source of the prostate, the source of the prostaglandins gave you that conflicting name. All right. So now you know that it's coming from the seminal vesicles. What role do they play? You see, they provide immunoprotection. Remember, we said that even in the male, within the male structure, sperms must be protected. Why? Because it is genetically foreign to the body. Because we said that the sperm cells has a chromosome listen of one N. Meanwhile, the body cells has 2N, and therefore it makes the sperms foreign. Now, the sperms are protected in a manner that they don't have any access to the bloodstream. Okay, and this we have highlighted that already. Once it also gets into the female genital tract, it's also going to be considered as what? As foreign. Because sperms, like I've said, wherever they found themselves, genetically they are different. Okay, so the vagina will also try to attack it as a foreign entity. So it is these prostaglandins that tend to protect the sperm cells. Otherwise, they will also the vagina system will also attack it. Okay, so prostaglandins are there to protect the sperms, especially within the female. All right. Again, there are a lot of.
I have a question.
Yes, sir. Please.
I remember during our previous interactions, you mentioned about autoimmunity.
Yes, sir.
So my question is, in that of today has to do with immunity from the female vagina. So
Yes.
in the male autoimmunity, is
in
it
the
the same
men's own?
prostaglandin?
Okay, sorry. I have to let you land. So land properly.
Oh, the last bit of the question was in the autoimmunity state, is it the same prostaglandins that are supposed to have offered protection for the male?
Sorry, no. You see, in this one, prostaglandins, we said they are coming from the seminal vesicles. Okay, so they are offering protection for the sperms in the female, but from the male itself, we talked about the blood tecido barrier. You remember?
Yes.
Blood tecido barrier, which will protect, also provided by the Sertoli cells, to ensure that there is no connection between the lumen of the semiferous tubules and then actually the vessels. Okay, so over the protection is not by prostaglandins. After all, we said that prostaglandins, they are not produced from the testicles.
Sure.
Okay, the prostaglandins are not, so they could not be the agent of protection for the sperm cells.
All right.
Okay, but they become agent of protection for the sperm cells within the female vagina. Okay. All right. How that is done, that is too complex, but just take that they offer a sort of form of protection. It's like trying to deceive them, the female immune system from recognizing the sperm cells as foreign. I hope that point is clear.
Yes, please. Thank you.
So all that you're saying is that this is supposed to be coming from the seminal vesicles. So all that you're saying is that the sperm cells are important. But if you have a failed seminal vesicles, it is, I mean, it's the same thing as not even having sperms at all, because whatever sperms you produce will not be able to go and deliver or do the work that it's supposed to do. All right. Now let's have some little information about prostaglandins. You said that they are coming from the seminal vesicles, and of course they become part of the seminal fluid. Okay. Now they help regulate the smooth muscle contractions, inducing peristaltic contractions in the female genital tract to facilitate the migration of the sperm from the vagina into the uterus. What happens here is that we know that sperms are supposed to just be deposited in the cervix area, and they are supposed to move into the uterus, to the cervical wall, to the uterus, to the fallopian tube, blah blah blah. Because the main concept is for them to go and to fertilize. Now, the cervix is almost always intact. Okay. Now, so once the sperms, the semen is produced and then locate, let's say, pushed into the cervix area during the sexual intercourse, what happens is that as we move along, you understand a certain concept of coagulation and decoagulation of semen. That's why we say that the semen has some semblance with the blood, because they also go through coagulation. Now, the semen will coagulate. And as to how it coagulates, we'll talk about all those things. It coagulates because we said that the environment, the vaginal environment, is highly acidic, and therefore highly what? Unfriendly. It provides an unfriendly environment for the semen. So once it coagulates, or it coagulates, what happens is that the semen becomes one bulky thing, not spreading within the vagina. Once it spreads through it spreads within the vagina, then whatever it is, the vagina acidic environment will overwhelm the alkalinity of the semen, and therefore kill all the sperms. But if it coagulates, what happens is that the vaginal environment can only have effect on the outside of the gel. I want you to imagine it. If there's a gel, the whole sperm, the whole semen which has been produced forms a clot. Then you realize that it's only the outside of the clot which will be affected by the acidic environment. Do you follow that?
Yes, please.
But the inside will be intact. The inside. So a lot of the sperms which are inside will be protected. That's why as we move along, you realize that, oh, semen, the sperms have to be a lot, in millions, because some of them are necessarily going to die. They are going through an environment which is hostile, and therefore some of them will sacrifice. But once some of them are sacrificed, those who find themselves within the clot will be protected. Now, so what happens is that once that happens, then the prostaglandins will soften or cause a smooth muscle contraction. So the uterus, sorry, or the cervix will be softened because of the prostaglandins, which acts by contracting the smooth muscle of the cervix. So it makes it softer. Once it makes it softer, meanwhile, there is a decoagulation process also taking place within the clot. And therefore, with little time, the semen will clot out. Sorry, will decoagulate. It first had to coagulate to protect it. Now, of course, the outside, those who are outside will all die, but those inside will be protected. Now, the decoagulation process is taking place within the gel, and then they will meet again. Once they meet again, meanwhile, the prostaglandins in it is also working on the cervical wall, the cervix, making it softer, so that now it will now give way for the sperms to move through. You see how it works. Now, this thing, even before science came in, our forefathers, who were even illiterate, knew that sperms can act as inducing labor. So if you have a woman who has been pregnant for over 9 months, sometimes they become pregnant for even 10 months, and let's say even they are not delivering. The old men will tell you that go and have sex with your man. You know what happens? You know that once you have sex, you are going to, even though it may be difficult, if you have a pregnant woman who probably after 9 months is not the same, they could have a challenge even, but the man should have an experience how to have sex with a pregnant woman. How many of you have had sex with pregnant women before? You don't go on top of. You should know the back way out. So if you have sex, what happens is that the semen will work as we're talking about. They go into the cervical area and then soften. So they say it wraps the cervix because it's going to, the prostaglandins in there serves to cause the smooth muscle also to contract. And therefore, once the smooth muscles are forced to open up, and then the cervix, I'm talking about the cervix, open and close, what it means is that it is inducing labor. And like I've said, the old men who didn't even go to knew about this one. That if a woman is refusing to deliver, or it's not labor is delayed, they'll ask you to go and have sex. That one, the old men know it. All right. And this is the concept behind it, that the prostaglandins in the semen play a role of wrapping the cervix, making the cervix like flexible. So once a pregnant woman's cervix becomes flexible, what happens? You are inducing labor. And that has a useful role to play in. That's why I'm saying that the prostaglandin play various roles in the reproductive process. Here you're talking about reproductive process, especially with respect to the female. Is that point clear, please?
Yes, please.
Right. So we are still coming back to the seminal vesicle secretions. We said, so we talked about the fructose, we talked about the alkaline nature, we talked about the prostaglandins, we talked about the important role of prostaglandins even in labor or parturition. All right. So we talked about seminal vesicle secretion also being rich in clotting or coagulation factors. The main coagulation thing is the fibrinogen. Contains a lot of fibrinogen. All right, and we know that from the hematological concept, the clotting mechanisms. You have it. Finally, you have the fibrinogen being converted into fibrin, and the fibrin, like thread-like entities, which will now entangle the red cells to form a clot. So the same thing happens within the within the semen, and so you see that once it's like something cuts you, you have a cut and blood is coming out. You see that the blood is coming out just within a matter of time, just a few minutes, it will clot automatically. The same thing, semen being produced. Once semen produced, within the next 5 minutes, it should clot. Okay, it should clot. And once something is clotting, means that there are clotting factors. And these clotting factors are also coming from the seminal vesicles. And the key clotting factor here is fibrinogen. All right. So this concept, you must understand it, that once you get semen produced, it will clot through a lot of mechanisms. And of course, once it's going to clot, there must be a clotting factors. Where are the clotting factors coming from? They're coming from the seminal vesicles. So if you have semen not clotting, where or the semen is watery, you get semen in the lab and it's watery, it doesn't clot. What does it mean? It means that there's a problem with the seminal vesicles. Okay, now what is the import of semen being clotting? Why should it clot? Like I've told you, semen, the sperms are going to go to fertilize. All right, a female egg, and then leading to reproduction. Okay, now I've said over and over again that the vaginal environment is very, very hostile, very acidic. So and then the semen is supposed to clot to protect majority of the sperm cells. If it doesn't clot, and then the semen just goes in there, and then you remember that, okay, not to remember it, you may have heard that some women will tell you that, oh, I have a man once, even when you have sex, all the semen will come out. Have you heard that story before? Some women will complain that you have sex and then you see that all the things have come out.
Yes.
There's failure, and those women, they will tell you that they have fertility problem. Most of them, they will tell you that they have fertility problem because the semen, once it goes there, must clot automatically. It must clot. And I've said that the clotting has a principle that the environment, the semen, the vaginal environment is so hostile, it's acidic, very, very acidic. We know that vaginal pH of even 4.5, it is supposed to be, it is called an alkaline. If the vaginal environment is 4.5, it is supposed to be alkaline. Who tells you that 4.5 pH is alkaline? Yet, as far as the female vagina is concerned, the pH of 4.5 is considered to be alkaline. So it is highly, very, very acidic, always below 4.0. So if you have the semen going in there unclotted, they will all be killed. They will all be killed. So the principle I mentioned earlier, in fact, you're going to repeat some of these things when we are dealing with seminology proper. But I'm giving you this background so that once you get to seminology, you just brush through. Right. So the clotting effect is so important to protect majority of the sperms. And like I've said, if you haven't heard it, I'm sure as you practice, some will tell you that, oh, we know about that. So that way you should know, you should be able to know the challenge. All right. Now, if the sperm to clot and it doesn't also decoagulate, that's a problem because once it clots, what is happening is that it's entrapping majority of the sperms. All right, a lot of them will be killed. Those within the outside, they'll be killed. But majority of them will be within the clot. So if the clot doesn't, if it doesn't also clot, sorry, if it doesn't also decoagulate, then it will remain a clot there and then nothing will happen. So there must also be a principle, a mechanism to decoagulate. All right, to liquefy it. Now, so it means that there must also be a principle, some proteolytic activity, because the fibrinogen become fibrin is a protein, isn't it? So it must be a system to lyse this protein. So proteolytic, and that proteolytic this mechanism is coming from the prostate. Okay, so if you, and we'll talk about that one in due course. So if the prostate also fails to also function, what happens is that sometimes you have semen in the lab. It will stand there for about 3 hours. It will remain a clot. And once it remains a clot, it doesn't liquefy. You can't work on it. Okay, because it must liquefy for you to be able to even measure the volume, to be able to assess the motility, everything. Okay, so those are the concepts. Now let me say this before we move on, and we will deal with this one under seminology. I'm saying that most of the time, we don't observe the clotting aspect of semen. Why? Why? Because the clotting should take place within the first 5 minutes. And by 15 minutes time, liquefaction should have also taken place from 15 minutes. So, but remember that most of the semen are produced in the house and they bring it to us in the lab. All right. So, and invariably, the earliest they can bring you is about say 15 minutes or 20 minutes time. Okay, sometimes because some people bring it after 30 minutes. So within the first 5 minutes where you should have observed the coagulation, the semen is now being transported from the house. So by the time it gets to you in the lab, the coagulation process have already passed, and now it's decoagulation which is taking place, liquefaction which is taking place. So you are unlikely to see the coagulating and then the decoagulating principles. But you must understand it that naturally semen must clot naturally for the reasons that you said. And after it has clotted, within some few minutes, let's say 10, 15 minutes going, it must also decoagulate. It must liquefy so that the sperms can now move. Those who have been protected must now be able to move through the cervix, through the uterus, and then up and go. So those are some of the mechanisms which you must appreciate. Once you appreciate that, you will see once it gets to seminology proper, it becomes easy, and then also semen analysis becomes even more easy. All right. Okay. So there are other proteins. We are mentioning this because if you are lucky to find yourself in advanced places where you have everything, then you know that, oh, if you want to assess the functionality of certain accessory organs, you can test this, you can test that, you can test that, you can test that. All right. You don't have them here, but that doesn't mean that you shouldn't know them. You should know them so that once now you have the chance, you'll be able to work on them. That after all, you know that. But when you talk about seminogulins, you know them, that these are the proteins that are also found in semen, specifically produced by the seminal vesicles. So if you can detect seminogulins, then you know that, oh, you want to assess the functionality of the seminal vesicles, so to speak. That's why we are mentioning all these things. All right. So we have proteins like fibronectin. Remember, we have the fibrinogens, which are for the clotting. We also have fibronectin and seminogulins. Seminogulins, you have seminogulin 1, seminogulin 2. They are the dominant protein components of the seminal fluid that causes semen to become sticky and jelly-like. You see, like we said, the semen, when you're talking about semen, you must also talk about their viscosity. So their viscosity is primarily also coming from the seminal vesicles. So if the semen is watery and so forth and so on, you know that necessarily the problem is coming from the seminal vesicles, because they must produce seminoglycans, which will give you the gelling or the jelly-like nature of the seminal fluid. All right. Now, seminoglycans 1 is supposed to be sperm motility inhibitor. Remember, we said that the semen, the sperms must initially be immobilized, one through the clotting, and then through also the seminoglycans. The clotting will clot it. But if within the clot, the sperms are struggling to move, by the time they are released, they will be dead. Okay, so the seminoglycans is supposed to keep them while the thing is has clotted, to keep the sperms immotile, so that you know that at this point we don't have to move, we don't have to waste our energy moving. Okay, so as they decoagulate, the seminoglycan level will go down, the sperms now becomes activated for them to move. So those are some of the natural principles behind the functions of the sperms. There are also a lot of other seminal vesicles contents. Seminal vesicles also contain acid alpha glucosidase, acid alpha mannose, beta GGTs, and then 5-alpha reductase. 5-alpha reductase is important here because remember we said that the seminal vesicles will need testosterone to function properly. All right. Testosterone now produces 5-alpha reductase, and it will metabolize the testosterone into dihydroxy testosterone. So you see that some doctors will request for DHT. Okay. Okay. So we have the 5-alpha reductase, which will reduce the testosterone into forming the dihydroxy testosterone DHT, which is more potent. All right, more potent than the ordinary testosterone. All right. So in all, we're saying that the seminal vesicles role is so critical. You can have your sperms produce all right. Seminal vesicles, if it doesn't also function, you are only producing sperms for nothing. They can't go and do anything. All right. So that is for the seminal vesicles. Any question, please? All right. So quickly you move to ejaculatory duct. That's a short one. Because from ejaculatory duct we go to the prostate, because the prostate is more the step there is much. We will push prostate to tomorrow so that we have more time to discuss it. So ejaculatory duct is very simple. You finish with that one, and then you postpone our lecture for today to tomorrow to be able to complete the prostate. After the prostate, you are done with the module 1, and then God will Thursday you will start with module 2. Any question, please?
Okay, so
yes, sir.
This is Alex.
Yes, sir.
You were talking about some ladies complaining of when the sperm is being given to them, it comes out.
Yes, sir.
And you were saying whether the problem is coming from, I want to know, in that circumstances, the problem is coming from, is it the lady, or because it's watery, we already know that is the man?
Oh, we are talking about the semen. We are saying that the semen, the fluids that accompany the sperms, they play important roles to protect the functionality of the sperms themselves. Okay, we are talking about the role the seminal vesicles play in this protection, and we're saying that one, first and foremost, they contain fructose. And the fructose is to ensure that the sperms, for as long as they remain stored, or even while they are being ejaculated, they need to have energy to move. That their energy source is fructose. The fructose is coming from the seminal vesicles. So that is one of the key things that will be required for the sperm viability. We also said that they are also alkaline. And we're saying that the alkaline is very, very critical because the environment where the sperms are going, which is the vagina, is highly acidic. So then, even before the vagina, you're talking even about the urethra, because the sperms must pass through the urethra and come out. All right. The urethra is also naturally acidic because you have urine passing through the urethra all the time. And urine is, as much as possible, acidic. So once urine is acidic and is passing through the urethra all the time, then the environment of the urethra will automatically be acidic. Is that also a point? So if the sperm is coming to pass through the urethra, and the sperms are cannot survive acidic environment, then even within the urethra, they can be killed. So you need an overall alkaline pH to be able to neutralize the urethra. Okay, so that is that. And you said that the vagina, what do you call it? Lining, the vagina wall, in fact, the cavity is so acidic. And I'm sure some of your colleagues rightly mentioned that the pH of the acid, of the vagina is around 3.8 to 4 maximum 4.5. Okay. Yeah. So how acidic it is. So you must have a higher pH, which is alkaline, to be able to neutralize that as much as possible. That is why even, you see, if semen volume is also low, if somebody produces very scanty semen, it is also likely that because the semen volume is low, that low volume is unable to produce an acidic, so alkaline enough to be able to neutralize the vaginal pH. Do you get the concept?
Yes, please. I get it.
Once you understand all this, when we are talking about male infertility, because we get to a point where we'll be talking about male infertility, and all this is coming. Somebody who produces, let's say, not oligospermia.
Hypospermia.
Low volume.
Hypospermia.
Very good. That's very fantastic of you. So hypospermia, that's low volume, not if it's low volume, the effect is that because it's low volume, their overall alkaline pH will not be enough to neutralize the vaginal environment. And therefore, all the sperm that we get in, they will all be killed. Okay, so we talk about, and then we came to talk about a very critical, important aspect, which is the clotting factors, also produced by the seminal vesicles. The clotting factors, their role is to make sure that once you ejaculate, the sperm will clot. Okay. Once it clots, what it means is that it is protecting majority of the sperms. Do you get that concept?
Yes, please.
I say prior way. So once it clots, what it means is that once it has clotted, majority of the sperms will be entrapped within the clot. And therefore, the environmental effect for the vagina will be very minimal and to be restricted only to the surface of the clot. Is that not it?
Yes, it is.
So if there's a failure of clotting, it means that the seminal vesicles is not producing what it's supposed to produce. And that means that the semen which will go into the vagina will be will not clot. And once it doesn't clot, what it will be liquefied. Once it liquefies, what do you expect it to do? It will come out. Even if it doesn't come out, all of them will spread within the vagina and all of them will be killed. So you understand these fundamentals. Once we get to the semen proper, you talk about all these things, and then you can link it to male infertility. What causes male infertility? And you agree that some of them, hypospermia, when the low volume ejaculate, will lead to. Why will it lead to infertility? Because the small amount of volume which is coming out will not be enough to neutralize the vaginal environment, and therefore sperms will always have a danger of acidic attack. Is that all right?
Wow.
So what we are saying here is that in the lab, if you have been working with semen a lot, you can have you experience semen which is like watery. Okay.
Yeah.
If it is watery, then it means that it couldn't gel. Remember that apart from even the clotting, we said that the seminal vesicles will also produce seminogellins, which gives you that kind of gelling effect. So if the semen is watery, it means that seminal vesicle has a problem. And we are going to deal with conditions that can affect the functionality of the seminal vesicles in our next round of conditions and effects that affect the reproductive system. Is that point okay?
Yes, please. Thank you very much.
You're welcome, sir. So quickly, we get into the ejaculatory duct. Very simple. It's also called ductus ejaculatory. It can also be ejaculatorious, where the end become RIUS. Once it is RIUS, you are talking about one ejaculatory duct. Remember, ejaculatory duct is also a paired structure. All right. You see, within the reproductive system, certain structures or organs are paired. Some of them too are not paired or single. You must know those ones which are paired and those which are single. So epididymis, the testes, the vas deferens, the ejaculate, all of them are paired. All right. So when you are talking of the plural, for example, ejaculatory ducts is plural. So that ductus ejaculatory becomes the plural form of the ejaculatory duct. If you want to single out one ejaculatory duct, then the ejaculatory becomes ejaculatorious, RIUS. I hope that point to just this one just for grammatical whatever gymnastics. All right. So each ejaculatory duct is formed by the union. I've said this already in our previous lesson. The union of the seminal vesicle excretory duct and the ampulla of the vas deferens. I hope that you can recollect that the vas deferens, the terminal portion is enlarged to become the ampulla. All right. So, and then the seminal vesicles, which we'll be talking about, has a duct. And because the duct is excreting something, bringing out the fluid, the duct can be said to be an excretory duct. All right. So this excretory duct combines with the ampulla or the vas deferens, that become the ejaculatory duct. The ejaculatory duct pass through the prostate before opening separately into the prostatic urethra, providing a pathway for the semen to travel to the outside during ejaculation. This is very simple to understand. That the ejaculatory duct, right? The ejaculatory duct, seminal vesicles, the vas deferens, here is the ampulla, they join, so this becomes the ejaculatory duct, this portion, and the ejaculatory duct. You know that the ejaculatory duct is actually passing through the prostate, all right? And then over here, you have the opening, the ejaculatory duct opening into the prostate, the urethra within the prostate. You know, that one also prostatic urethra. All right. So these are all some few pictures for you to appreciate it. It's very, very simple. Very, very simple. I don't want us to start the prostate today. Prostate is more elaborate. And so you deal with the prostate, God willing, tomorrow. Once you're done with the prostate, you will be done with all the structures and organs that make up the reproductive system. And like I've said, again, we move to module 2. Module 2 is now going to talk about the phenomenon, some of the phenomenon that affects the parts. All right. For example, if you talk about a penis, then one of the major simple phenomenon is erection. Okay, so you're going to deal with erection extensively. You're going to deal with the types of erections, and then what we call postmortem erection, or erection is the same as tumensis, and we're going to deal a lot. So after that, we talk about ejaculation and all have you, and then talk about the diseases that can also affect the penis, talk about diseases that can affect the testes, and a whole lot of. So as we've done with them individually, we're also going to deal with the conditions that affect the individual structures and organs. Because once you understand all those things, then if it comes to diagnosing male infertility, you know where to look at. You know what is likely to give you what. All right. So, ladies and gentlemen, today's has been short, but I don't want to start something which we cannot complete. The process is very, very elaborate. So we'll deal with that, God willing, tomorrow. All right. Any question, please? Is there any question anybody want to ask?
No, sir.
Okay. All right. Thank you for your time and attention. And I want to believe that you are learning something.
Yes, please.
Sir,
All right.
sir,
Yes, sir.
this is Alex. So that smell that the semen is having, is it also coming from the seminal vesicles?
The what?
That
The
smell
smell,
is
the odor.
The smell.
Is it also coming from the seminal vesicles, or that's part of the 30%?
You will talk about that tomorrow.
Okay. Okay.
All right.
All right.
Okay. Any other question, please?
Grandpa.
Yes, dear.
Yeah, this is Rhoda. So
Yeah.
you mentioned that the volume also counts when it comes to sperm surviving the acidic environment. But there have been instances where some individuals who produce a semen sample, the volume is low, but the sperm count is very high. So in that case, does it mean that the chances of majority of the sperm surviving the vaginal environment is low, considering the volume which is low?
All right. Thank you very much. I've said that all these things are fundamental information for you to appreciate or understand better when you're dealing with seminology. We haven't gone to seminology. If you get to seminology, all these things will come out. And remember, we are now going to talk about ejaculation. And you see under that topic that the semen that is brought to the lab a lot of the time doesn't represent the entirety of the seminal volume, because most people are unable to produce semen by masturbation, and they produce semen through what we call coitus interruptus. You deal with that. Coitus interruptus is, let's say you are having sex, they say go and bring urine. For example, they say they go and bring midstream urine. Midstream urine means that allow the first flow of the urine to pass, collect the middle portion, and then still throw off the last portion. That is midstream. And midstream is used for a specific purpose. You also have, let's say, terminal urine. If you are suspecting schistosomes, you don't need midstream urine. You rather need terminal urine. In that case, it will tell you that allow the urine to flow as much as possible. When it's about to finish, push the last portions into the container and bring it to me. That is the terminal urine. And that's where you are likely to get your schistosomes if they are present. Sometimes somebody will tell you that, oh, but this person produced the urine without the procedure that you're talking about, yet I got the schistosomes. If the person has heavy infestation with the schistosomes, then no matter how urine is collected, whether it is the early urine or midday urine or terminal urine, whatever, because the extent of infection is too huge. Any amount of urine that is coming out, you get some of the. But you see, if you have malaria 4 pluses, MPs, slide, and 1 plus, which is more significant? Malaria 4 pluses. Malaria 1 plus. Malaria slide 1 plus. Malaria slide 1 plus and 4 pluses. I'm saying malaria 1 plus slide and malaria 4 plus slide, which is more significant? All right. As far as I know, if you have malaria 4 pluses, even a first-time laboratory science student will see it. Okay, if you have malaria 4 pluses, nobody will miss it. Everybody will just see it. Malaria 1 plus, you must scrutinize. You must be that experience to be able to pick 1 plus. Okay, and that's more important, because if the 1 plus, if you miss it, that 1 plus will now become more severe infection and cause severe damages to the patient. So to me, if you detect 1 plus, I think that you've done more work than the person who did the 4 plus. 4 plus, wouldn't any boss slide, so I was with the MPs. Okay, but you can't do that for 1 plus. 1 plus, you must scrutinize, you must look through the slides everywhere before you see a parasite, 1 or 2. Okay, so all in all, I'm just trying to connect something, that the semen which comes into our lab, all right,
Okay.
you prepare the patient, and then you advise the patient to produce the semen by masturbation. We will talk about these things when on the seminology proper. Why? Because 1, semen or ejaculation, like I said earlier, is fractionated. It comes in fractions. All right. If the person tries to do what we call coitus interruptus, where coital means sex, interruptus means just interrupting, so that during the action, during the process of sexual intercourse, the man will get a feel that is about to reach orgasm. And therefore, at that point, we expect you to remove your penis and then ejaculate. That's what we call ejaculating inevitability. Ejaculating inevitability is that when you say something is inevitable, it means that it will come, no matter what. So once you reach the orgasm, if you remove your penis, no matter what you do, the ejaculate will come. Ejaculation will take place. But people think that, oh, they don't know that information, so they think that they want to see that, oh, they are giving off. I mean, the thing is not coming. By the time that you move off, the first portion of the semen is already gone. So by the time the person withdraws and then pushes it into the container, about half of the semen volume is already gone. Okay, so the amount of volume that we normally get in the lab is not representative of the entirety of the semen volume. And we deal with all that one when it comes into seminology. All right, but a lot of people also find it difficult to masturbate. Sometimes they give you excuses. Oh, my religiously, culturally, I'm not supposed to blah, 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 blah. They'll give you excuses. Someone will tell you, I haven't done it before, but you try to give the person the benefit, try to say that for the purpose of the test, you try it for us. You must be able to explain that during the sexual distance, which many people, even the lab scientists themselves, will tell you, oh, go and have sex if yours, oh, bad, then you know, a lot of that is a wrong procedure for semen collection. All right, we talk about all those. So yes, sometimes a small amount, but say the count is high. But you know, the count can be, somebody can produce semen of more than 300 million. So if some have passed, and you get 30 million, you still know that the person has normal semen. But in actual fact, if you had the entirety of the semen, the count could have been around 150 or 300 million. Okay. Now, consider a person whose semen count is around, let's say, 30 million. Normally, you will have gotten 30 million. But he produces semen in the manner that we described, where a large portion of the semen has gone. And as you know, I've said that semen is produced in fractions. They come in fractions. First fraction, okay. Second fraction, third fraction. Now, the first fraction contains about 70% of the sperms. So if that portion is gone, unknowingly, and the person's actual count is around 30 million, and then you now take a semen which has been produced by Quatis interruptus, and the person gets, let's say, 80 million. You say that the person is oligospermia. So those are you, and I'm saying that all these discussions is not under seminology. I'm intentionally bringing some of them in. So when you get to seminology, you will now understand them in full and appreciate them in full. And then you know that, yes, you have learned something. Okay, so sometimes I don't want to go too deep, but what I'm saying is that the seminal birth skill role is so critical and important. And they control the volume, they control the pH, they control the survival, viability, and so forth and so on of the sperm cells. And, I'm sure that as you move along, some of these things will come out very well for you to understand them. And I want you to understand them in a manner that even if you don't read, you understand. You should be able to remember some of these things and then support your understanding of the whole concept. Is that okay?
Yeah, thank you.
So the volume being small, normally it is unrepresentative of the actual semen volume coming from the patient. All right, we'll deal with that as we move along. All right. Any question, please? Any question? The woman, even though you don't have what we have, but you necessarily will have husbands, you necessarily will have children, you necessarily will have brothers. So your understanding of this one is also very, very important. And as lab scientists, automatically you'll be doing seminal fluid analysis, which ultimately is our focus. But you see that the understanding of some of these things will help you to appreciate the seminal analysis in a broader detail. All right, on that note, I think we can bring today's session to a close and hope to see you tomorrow, finishing the prostate. Once we finish the prostate, we also end there and then continue on Monday with the full what we call physiological phenomenon of the female, of the male reproductive organs. All right, good night. See you tomorrow.
Good night.
So now, yeah,